So, I am Toshio Tsubota, the section chair of WDA-AP, Wildlife Disease Association, Asia Pacific section, and professor of wildlife biology and medicine, Faculty of Veterinary Medicine, Hokkaido University, Japan. I am very glad to hold 2024, the annual WDA-AP webinar tonight. I would like to introduce a little bit the WDA-AP activity before starting the webinar. WDA-AP was founded in 2019 as one section of Wildlife Disease Association, WDA. WDA is the international association with more than 1,500 members, and its mission is to promote healthy wildlife and ecosystems, biodiversity conservation, and environmentally sustainable solutions to One Health challenge. WDA-AP supports annual conference of ASCM, Asian Society of Conservation Medicine, every year, and the latest conference was held in Mongolia this month. You are very welcome to join the WDA and WDA-AP activity and events. Today, it is a great pleasure for us to invite two experts on elephant TB, tuberculosis, as presenters in the webinar. I hope the webinar will be fruitful and enjoyable for every participant. Thank you. So, firstly, I will introduce the first presenter, Dr. Susan Mikota. Dr. Susan Mikota is co-founder and director of veterinary programs and research at Elephant Care International, a non-profit organization dedicated to the health, welfare, and conservation of elephants. She has written numerous scientific articles and book chapters, including Biology, Medicine, and Surgery of Elephants in 2006, the first modern veterinary textbook about elephants. Dr. Mikota has lectured, presented workshops, or conducted research in Nepal, Myanmar, Indonesia, India, Thailand, Vietnam, and Sri Lanka, and has worked to improve the healthcare and welfare of elephants throughout Asia. She was awarded a Guggenheim Fellowship for her work with Sumatran elephants. Dr. Mikota is a member of the IUCN Asian Elephant Specialist Group, a diplomate of the American College of Animal Welfare, and adjunct professor at Michigan State University. She is based in Tennessee in the United States. The talk of Dr. Susan Mikota today is about overview of tuberculosis in elephant and prevalence in Asia. So if we are ready, Susan, you can start now.
Okay, let me get everything set up. Okay, you can see my slides?
It's fine.
Okay, all right. Well, thank you, Dr. Tsubota, for that introduction and for inviting me here today to talk about TB in elephants. So these are the main topics I'll be presenting. I've got a few introductory slides, and then I'll talk about clinical signs, how TB is diagnosed, TB treatment, and I'll discuss the studies that have been done in Asian elephant range countries that give us an idea of the prevalence of this disease. And I'll close with a brief discussion of zoonotic issues and some conservation concerns. TB has been a disease of animals and humans for millennia. Knowledge of the disease in humans dates back thousands of years, with the earliest written records out of India over 3,000 years ago. And there's been a wasting disease that was described in elephants. It was probably TB, and it was mentioned in ancient Sanskrit documents. But it wasn't until 1834 that the disease was named by a German physician. And the causative agent wasn't discovered until March 24th, 1882 by Dr. Robert Koch. And March 24th is now designated World TB Day by the World Health Organization to raise public awareness of the ongoing global crisis of TB in humans. TB is now the second leading cause of death by an infectious agent after COVID. Nearly 4,500 people die and 30,000 people fall ill every day. And then yearly deaths exceed 1 million. Now, TB in humans has been scientifically studied for about 150 years, and yet there is still much that is not understood. The scientific study of TB in elephants has occurred only for a few decades. Before I turn my focus to Asian elephants, I want to briefly mention some other wildlife species that have been impacted by TB. Since 1990, African buffalo in Kruger National Park have been under surveillance for TB. TB, now caused by M. bovis in this case, has spread from the southern to the northern herds in the park, and it's spilled over into other mammalian species. Currently, there are 25 wildlife species, rhinos, cheetahs, lions, and buffalo. We have not seen a similar event, but I think to take TB seriously wherever it occurs in wildlife populations. Now, there's even some evidence, although it's controversial, that TB may have been a hyper disease and it may have played a role in the extinction of the mastodon. This researcher examined mastodon skeletons, and he found TB lesions in the foot bones and the ribs in 52% of the 130 skeletons that he looked at. And the foot lesions were identical to those that are considered pathognomonic for TB in bison. Now, the author didn't suggest that TB alone was responsible for extinction, rather that TB may have weakened the animals and made them more likely to be killed by predators. So a few facts about TB in Asian elephants. As you know, it's a bacterial disease. And in elephants, most cases of TB in Asian elephants have been caused by M. tuberculosis, the human strain. For reasons I will explain, diagnosing TB in elephants is challenging. Treatment is difficult, and TB is a zoonotic disease that affects both humans and other animals. It's a respiratory disease. In humans, TB is spread on respiratory droplets during activities like coughing and sneezing. Elephants can contract TB from humans, but once they're infected, TB can spread from elephant to elephant and also back to humans. In Asia, where elephants intermingle with cattle, they might also become infected with the bovine form of TB, but this hasn't really well been studied, and I'm not aware of any published reports. Clinical signs, like those that occur in humans, may also be seen in elephants. Things like weight loss, respiratory discharges, coughing, exercise intolerance. However, TB is a chronic disease. It develops over a period of years. Many elephants do not show signs until the disease is quite advanced. 1996 is the year that we became aware of TB in elephants in the US. These two circus elephants died only 3 days apart. This created a great deal of public concern, and an advisory panel was formed by our US Department of Agriculture. Now, both of these elephants have had TB. You can see that the elephant on the left is very emaciated, but the body condition on the right is quite normal. Diagnostic tests for TB can be classified as either direct or indirect. The direct methods are actually detecting the TB organism. And these methods include culture, acid-fast stain, and nucleic acid amplification tests like PCR. Indirect tests are typically immune-based, so they are detecting some change that occurs in response to the presence of the bacteria. The intradermal skin test, as an example, is measuring cellular reactivity against mycobacterial antigens. Serological tests detect antigens or antibodies, and I'll be talking more about serological tests later. Culture is the gold standard for the diagnosis of TB, and while culture and other direct tests may be preferred because they are actually detecting the TB organisms, the level of detection differ. For example, for TB to be detected using acid-fast smears, a minimum of 1,000 organisms per ml must be present. And the acid-fast smears are not specific for TB. Other organisms such as nocardia are also acid-fast, as are many of the non-tuberculous mycobacteria, which are commonly found in elephant respiratory secretions. So in these cases, species identification requires confirmation, either by culture or DNA probe. To be detected by culture, 100 organisms per ml must be present. Nucleic acid amplification tests can detect as few as 1 to 10 organisms, but there are some inherent challenges to this test as well. So unfortunately, we don't have a lot of options to diagnose TB in elephants, and the methods that we do have all have limitations. We can't X-ray elephants. Clinical signs alone are not adequate, as I showed with those 2 elephants in the previous slide. And the intradermal skin test is not accurate. It has a sensitivity of only 16%, a specificity of only 74%. Identification of the causative agent is, of course, ideal, but this has been problematic in elephants, which I will explain. So a great deal of work is focused on indirect tests, mainly serological assays. So how do we collect a trunk wash or a respiratory sample from an elephant? The procedure that's been developed is a trunk wash, and it's comparable to a sputum sample collected from a human, although it's not as easy to collect. Elephants should first be trained to allow their trunks to be handled. Sterile saline is instilled, the trunk is elevated and then lowered, and the sample is collected in a plastic bag. Well-trained elephants will forcibly exhale so that the sample is from the lower respiratory tract, and that's desirable. However, not all elephants comply. Many captive elephants in Asia do not allow their trunks to be handled, or they haven't been trained to perform a proper trunk wash. So because of this, there's a modified trunk wash procedure that has evolved in India and Nepal. The trunk wash is self-administered by the elephant aspirating saline from a bucket and then either passively or actively blowing the saline back into the same bucket. Now, the quality of the sample obtained by either trunk wash method is questionable because we're essentially trying to obtain a sample from the lungs through a 2-meter-long pipe. So what are the limitations of culture? Failure to isolate the organism does not rule out infection. And that's because intermittent shedding is common in elephants, so there's a potential for false negative results if the elephant was not shedding on the day the sample collected. Now, because elephants use their trunks for such a variety of functions, contamination is a common problem. If the sample's overgrown by other bacteria, then the results are non-diagnostic. Reporting time is slow, and culture requires 3 samples from each elephant. That may not be practical or affordable to screen large numbers of elephants in Asia. Human labs, particularly in TB endemic countries, may be unable or unwilling to accept samples from elephants. There are some extra decontamination steps that are recommended, and labs that are already overburdened by a heavy human caseload may not be able to accommodate this. So this slide illustrates why culture is problematic. In Sweden, they collected 189 trunk wash samples from 5 elephants confirmed infected at postmortem. only 6 of the 189 samples were positive. In Thailand, only 2 of 60 samples were positive from 4 infected.
ไทยแลนด์เนี่ยเจอ positive
elephants were
สอง
sampled.
ตัว
Only 1
อัง
of
กฤษ
371 samples were positive, and in Switzerland, they collected over 200 trunk wash samples from 10 elephants. All of these 200 samples were negative, but 3 of these elephants were confirmed infected at postmortem. So I think you can see that while a positive culture confirms TB, a negative culture does not guarantee that an elephant is TB-free. I'm going to be focusing on serological studies, but I wanted to make everyone aware that there has been some work done to develop gamma interferon tests for elephants. I've listed some of the references here, and if anyone would like PDFs of these publications, please send me an email. So most of the serological studies that have been done have used the elephant TB stat pack or the DPP vet TB test. These are both immunoassays, and they're aimed at detecting antibodies to specific mycobacterial antigens. The stat pack was licensed by the US Department of Agriculture in 2007, and it was replaced by the DPP, which was licensed in 2012. The stat pack had a single test line that was a combination of antigens. The DPP has 2 test lines and a control line. Line 2 is a fusion protein of ESAT6 and CFP10, and reactivity to this antigen combination has been found in almost all of the MTB cases in elephants. Line 1 is the MPB83 antigen. This has been found mainly in elephants with atypical mycobacteria. Now, licensure of the DPP was based on 40 culture positive cases that were DPP reactive and 147 culture negative cases that were DPP non-reactive. It's an easy test. It takes only about 20 minutes, and it can be performed in the field using whole blood or serum. And this shows the possible outcomes of the DPP test. The test is reactive and most likely represents TB if test lines 1 and 2 or just test line 2 are positive. If only test line 1 reacts, it's considered reactivity to non-tuberculous mycobacteria. And if only the control line shows, then the test is considered non-reactive. And this slide just shows a close-up of the possible results. As I mentioned, test line 2 is a fusion protein of CFP10 and ESAT6. Test line 1 is MPB83. These are the 2 main references that describe the DPP. There are other publications as well, and I can make these available to anyone who's interested. So now what I'd like to do is share a few cases from Nepal that show the correlation between DPP results and confirmed disease that we have seen. Both of these elephants were bulls that were used to patrol the national parks. Elephant A was 55 years old. Elephant B was only 30 years. Both were reactive on the DPP test. In the bottom photos, you can see what their lungs look like at postmortem. This is another 2 cases. Elephant C was very old. She was over 60 years when she died. She was DPP reactive and under permanent quarantine because we considered her too old to treat. MTB was cultured from her lungs at postmortem. In 2 cases in Nepal, we have found that the DPP is reactive on other body fluids, which parallels some findings in the US. We did not have a premortem serum sample from elephant D, whose lung tissue is shown on the right. She died in 2009. MTB was cultured. 2 years later, we extracted fluid from the frozen lung tissue. We performed a DPP test and it reacted. So while the DPP was developed to test whole blood or serum, other body fluids may react. TB treatment was for elephants was without precedence when we began treating elephants in the US. So we used humans as the model. The elephant protocol is based on treatment regimens that we know are curative for humans. The basic protocol calls for 3 to 4 drugs for 2 months, followed by 2 drugs for 10 months. Isoniazid, rifampin, ethambutol, pyrazidamide are the first-line drugs that are typically used. Administration is oral or rectal, although rectal has some limitations. If at all possible, serum drug levels should be monitored and drug doses adjusted accordingly. Treatment details can be found in guidelines that we have posted on the Elephant Care International website. There have been a number of published studies on the pharmacokinetics of TB drugs in elephants. Again, we know we can achieve blood levels in elephants that work for humans. All of these first-line drugs that are listed can be given orally, and with the exception of rifampin, they can be given rectally as well. We've been unable to achieve adequate blood levels of rifampin using the rectal method. Ethambutol has been somewhat problematic when given rectally, as it can be rapidly expelled, but that may be related to the formulation. Ideally, we should have an accurate weight to calculate accurate drug doses. In Nepal, we use a portable scale that has 4 load cells placed under a very sturdy wooden platform. The weight's recorded on a digital readout. If a scale isn't available, there are a number of methods to estimate weight. We've used a method based on measuring the chest girth and have found it to be fairly accurate, except in very young elephants. But to determine proper doses for a disease like TB, a scale weight is preferred. Treatment has challenges. Some of them are listed on this slide. The recommended treatment course is 1 year. The drugs are bitter, and it's not always easy to disguise the taste well enough to overcome the exquisite taste receptors that elephants seem to have. There can be side effects, just as in humans. Elephants may become anorexic. Liver enzymes may elevate. Treatment's expensive. Drug resistance is a concern, and it has occurred in a couple of cases in the US. And unfortunately, we do not have a test that confirms cure. In Nepal, we initiated a DOT program for elephants. DOT is the acronym for Direct Observed Therapy, and it's a procedure that's commonly used in humans to enforce compliance. A healthcare professional actually observes the patient taking their TB medications. So that's what we did with elephants in Nepal. In this photo, Dr. Sarad Paudel on the left, Dr. Jiwan top on the right are counting pills for treatment. We've used, we've used 3 different administration techniques in Nepal, depending on what worked best for the individual elephant. Excuse me. In the method pictured here, the pills are placed in a ball of molasses or rice, or they can be hidden in bananas or other fruit. Even though the medication is placed in food, it's still given directly to the elephant, not offered free choice. Now, the second technique that we've used is direct administration. And that was discovered by chance with this particular elephant who was very suspicious of medications and food. Everyone was getting quite frustrated, trying to figure out a way to get this elephant to accept the pills. One day she opened her mouth and the mahout just tossed the pills in and she swallowed them. And she actually completed her treatment using this method. Because it is so difficult to get elephants to accept TB drugs orally, rectal administration is often the easier route, and most elephants are cooperative. Feces have to be removed first. The medications are crushed in water, administered using a funnel and long tubing. Expulsion can be a problem, but we have found that positioning the elephant on an incline for a few minutes after the drugs are given prevents this. And as I mentioned earlier, we can't give rifampin rectally because we do not achieve blood levels. Elephants that have poor body condition like this bull have improved with TB treatment. At the time this bull was treated, he was over 40 years old. He was very thin. We were afraid he might not survive treatment. Today, he's in very good condition, now in his late 50s. Had we not treated him, it is likely he would have died years ago. He may not be completely cured. We won't know until the very end. And because again, we don't have a test that confirms cure, but we can be confident that we've extended his life. So let's look at some of the serological studies that have been done that can help us understand status of TB among elephants in Asia. We have information from Thailand, Malaysia, India, Laos, Myanmar, and Nepal. In 2010, Dr. Tawi Pok reported the first elephant TB cases in Thailand, and these cases occurred between 2005 and 2008, and they were confirmed by culture and PCR. 3 of the 4 cases were serologically reactive on the stat pack. The 4th elephant had very advanced disease. The authors thought that she may have been allergic, and genotyping suggested transmission from humans. A 2013 study by Dr. Ong in Malaysia that also used the stat pack found a seroprevalence in elephants of 20% and a seroprevalence among handlers of 25%. And that was based on the quantiferin gold test, which is a gamma interferon test. PCR of the elephant samples yielded 3 that were positive for MTB from 3 elephants that were also seropositive. There were 7 seropositive elephants that had negative PCRs. Now, the negative PCR does not necessarily rule out TB. These elephants may not have been shedding at the time of sample collection. And we do not yet fully understand the stages of TB in elephants, how fast infection progresses, when shedding starts. We do know that the serological test can be reactive months to years in advance of detection by a positive culture. So expanding on the previous work, this was a prospective study, and it was aimed at identifying risk factors for TB in captivation elephants and their caregivers. 60 elephants at 6 different facilities were screened for TB longitudinally using the stat pack and the DPP assays, and 149 staff were screened again using the quantiferin gold. The facility records and questionnaires were used to mine information about potential risk factors. Seroprevalence was 23% among elephants, 25% among staff. The risk of infection was significantly associated with being a zoo employee or elephant handler versus a sanctuary employee that had less human or less elephant contact. And in elephants, the risk of seroconversion was significantly higher among elephants that were assigned mahouts with whom they had regular contact. In Laos, they examined 80 elephants and found a 36% seroreactivity using the stat pack. They didn't perform serology on the mahouts. However, 63% had at least 1 clinical sign suggestive of TB, and 8 had chest x-rays that suggested TB. 3 of the mahouts with the suggestive x-rays worked with serologically reactive elephants. And of these 8 mahouts, 7 had either a familial or a personal history of TB, 6 had clinical signs. Because TB is endemic among the human population in Laos and other parts of Asia, you can see how challenging it can be to draw conclusions regarding the association between TB in elephants and humans. So let's look at some of the work that's been done in India. In this study, Dr. David Abraham examined 473 elephants. He found an overall seroreactivity of 16.5%. And he found the highest seroreactivity among temple elephants, which have the greatest human contact, compared to elephants that are maintained by the government in forest camps, which had the lowest seroreactivity. And then the privately owned elephants were in between with a seroprevalence of 14.9%. To further look at seroreactivity of elephants in India, In this study, they took 179 samples. They randomly selected them from a group of 600 elephants, and they tested them using the StatPack and 4 newly developed ELISA tests. They did perform trunk washes on select StatPack reactive elephants. None were positive, but 1 StatPack reactive elephant was culture positive at postmortem. And overall, 15.9% of the samples were reactive on all 5 tests. There's been limited testing in Myanmar. In 2012, Dr. Kainu Mar and I tested 84 elephants, and we found no reactors on the DPP. In 2015, we tested 145 logging elephants, and we found 9 reactors. So from this limited data, it seems like the seroprevalence in Myanmar is low, but a wider study is needed. So we began testing elephants in Nepal in 2006. And to date, we have tested all of the captive elephants, except for the very youngest that have not been trained for bleeding. Many elephants have been tested several times using the StatPack or the DPP, and about 60 elephants have been treated. To my knowledge, Nepal is the only country that has a formal TB management program for elephants, but the program does need to be updated. And the ultimate aim of this program is to control TB at the captive-wild interface. A lot of what we know about TB in elephants has come from work that's been done in Nepal and also from Dr. Svoboda's lab at Hokkaido University. I've listed some of the recent publications, but in total there are over 50, mainly in peer-reviewed journals and also including at least 2 PhD theses. So to summarize the research that I've just reviewed, I think we can state that the seroprevalence of TB in the Asian countries studied is somewhere between 16 and 30%. Myanmar may be an outlier, but more study is needed there. Few studies have included culture, but it is important and more studies need to include it. The serological test can at least help us get a sense of prevalence, as well as guide the course of action for an individual elephant that's reactive. That may be additional serological testing, training for trunk wash, serial trunk wash collections, possibly segregation, even treatment, depending on the exposure history, other individual factors. TB is zoonotic, so what about human health concerns, especially now with the emergence of drug-resistant and multi-drug resistant strains? There have been numerous reports of human infection among elephant workers following exposure to TB elephants, mainly in the US. 1 active case has been reported, but it couldn't be determined if the elephant transmitted TB to the human or vice versa. It may be that the greatest risk is to mahouts and handlers who live in close association. However, whether TB transmission between elephants and humans occurs at public events like zoos, circuses, or large festivals isn't really known. To date, there have been no reports of elephant TB transmission to the general public, but no one has really studied this issue. The risk of TB may also extend to other species in the wild, especially in places like Chitwan National Park in Nepal. Government-owned elephants are used for anti-poaching patrols, for rhino censuses. Privately owned elephants are used for elephant-back safaris. Captive elephants are critical to the protection of the park, to tourism, to the local economy. Elephants, rhino, deer are all susceptible to TB. And you can see from the image on the lower right that the elephants get very close to rhinos during these elephant-back safaris. TB can also potentially be transmitted from an infected elephant cow to a wild bull when the bull comes for breeding, as commonly occurs in the elephant stables in Nepal. And you can see that on the image on the lower left. Certainly one of our greatest concerns is TB transmission to the wild. In 2013, the first case of TB in a wild elephant anywhere was reported in Africa. The elephant had been in human contact at an elephant orphanage before it was released. The next case in a wild elephant was reported in 2015 by Dr. Vijitha Pereira in Sri Lanka. And then in 2017, 4 more cases of TB in wild elephants were reported. The first article describes 3 cases. 1 case is reported in the second article. And both of these articles are online free in the March 2017 issue of Emerging Infectious Diseases. As we concluded in our first article, our results indicate that TB may be spilling over from humans, essentially a reverse zoonosis and emerging in wild elephants. Although we don't really know very much about the intersection of elephant TB with domestic livestock species, TB is nonetheless a One Health disease, and the overlap of elephant TB with humans, livestock, and other wildlife species needs further study. So to summarize, TB is a devastating disease for elephants. Our diagnostic tools are limited. Serological tests are a useful tool. It allows us to make more important decisions. And if we hope to mitigate TB transmission to the wild, these tests may be the ones that we need to rely on. As the previous director of the CDC said, if we wait, it is too late. So collaboration among veterinarians, human doctors, wildlife departments, private owners, all concerned parties is essential to develop appropriate guidelines to manage TB in elephants. And clearly more research is needed. While we have learned a great deal about TB in elephants over the past few decades, there is still much that we do not know, and research must continue. We need other diagnostic options. Collecting trunk wash samples is difficult. The quality of the sample is often poor. The test is expensive. So-called e-noses and breath analyzers that detect volatiles that are elaborated by the TB organism are under development in humans and cattle. We need to piggyback on these cutting-edge human research efforts to improve our diagnosis of TB in elephants. If validated, this type of testing method could allow us to test elephants more frequently and more economically. The expert MTB-RIF is a relatively recent technology, and it's becoming more widely used all over the world. It's an automated PCR that uses sputum, and it's a simple 3-step procedure that detects MTB as well as rifampin resistance in less than 2 hours. It has very high sensitivity and specificity, and it has been used with good results in several wildlife studies in South Africa. In the area of treatment, one of our most urgent needs is better ways to disguise the bitter taste of the medication. And right now, we give the medications orally and rectally, but what about inhalation therapy or transdermal application? We also need more pharmacokinetic studies, both for the first-line and second-line drugs. And we need some biomarkers for cure. I hope that these last few slides will inspire any young elephant researchers that may be listening today. One last thing, I've cited a number of studies in the presentation, and they are all included in a comprehensive list of TB publications on the Elephant Care International website. And as I mentioned earlier, I'm happy to share any of the PDFs that I have. Once again, I'd like to thank Dr. Sabota, the Wildlife Disease Association, webinar organizers, and I'd also like to acknowledge the National Trust for Nature Conservation and the Department of National Parks and Wildlife Conservation, both for supporting research on elephant TB in Nepal. Thank you for listening, and I'd be happy to answer any questions.
Thank you, Dr. Susan Mikota. Very nice talk. So we have time for question and answer. So if you have a question, you can do unmute or put the question at the chat box, please.
I have a question, if you can hear me.
Yeah,
Yes.
okay.
Hello, thank you for the great presentation. Because I have a project on wildlife disease in Africa, and I'm curious to know, so it's reasonable that why there are more human TB in Asian elephants in comparison to in Africa because of more human contact with elephants. But my question is, why there are not high loads of bovine TB in Asian elephants, because we know that bovine TB is the main problem for African elephants because of the contact with buffalo and other bovids. But in Asia, we have many local livestock ranches for local cattle, other local livestock, and they're living close to the Asian elephants. So why we don't have that high rate of bovine TB in Asia?
Well, it's a really good question, and part of it may be because there hasn't been a lot of research to look at that issue. You know, and I have some slides where I show elephants and cattle intermingling, but the associations that I've observed have not been prolonged. You know, they've been brief, elephants passing through a field where cattle are grazing. You know, that may be a factor. There may be some differences in you know, genetically in species resistance. I don't really know. In the US, there's of all the, we've had well over 50 cases of tuberculosis in captive elephants and only 1 or 2 cases of M. bovis. The one case that I recall was from a elephant that was housed with a rhino that had M. bovis. So it would be an interesting thing to dive into a little bit more deeply.
Thank you, because
yeah, I don't have a good answer for you. I'm sorry.
no worries. But because one of my friends, one of the colleagues I know from the UK, she was interested. She's project was on bovine TB in Malaysian Borneo elephants, mainly in Sabah. But I know that the Malaysian government didn't like that project. They actually they terminated the project at the beginning section of the project. I don't know why, but they said that we don't have this bovine TB in Borneo. So I know if you say that there needs to be more research, there needs to be more collaboration for this to be evolved.
Yeah, I think there does. You know, sometimes it's governments may be a little bit nervous about finding things, and at times it can be easier to not look. But I don't know, there could have been a number of reasons why they didn't want that project to go on.
Thank you so much. So you think there could be host differences between the susceptibility of Asian elephant in comparison to Africans?
Yeah, there could be, or again, it could be environmental, it could be exposure differences. You know, all that needs to be teased out.
Thank you very much. Thank you so much.
Okay, if you have other questions, send me an email. We can continue to ponder.
Yeah, sure.
Okay. Any other questions? We have 2 questions at the chat box. Susan, can you see the chat box?
Tamam abi dur şimdi bir şey yapıyorum ya.
I do. Let me scroll down. Let's see. Let's see. Oh, Arjun, to everyone, can you suggest any particular time to collect trunk wash samples? Because trunk wash samples show false negatives in many cases.
Mm-hmm.
I don't, you know, I know there are some parasites that show, you know, diurnal, you know, we have ability to detect them at different times, day or night. I don't know that that's what's going on with trunk washes. I think they're either shedding or they're not. I've followed a number of elephants here in the US, and one day we can do a trunk wash and isolate TB, and the next day we can't, and the next 3 days we can, and then we can't. It's quite variable. So it's a matter of shedding. It can also maybe the quality of the sample day to day. It just makes it challenging to try to get a diagnostic sample. Is there another question? That was the only one I saw.
Another question is, which one is the best tissue of choice in live elephant? I think
Which
for
is-
diagnosis.
Which is the best, the best,
Tissue.
oh, the best tissue of choice?
Yeah.
Oh, from postmortem, there's a postmortem protocol that recommends certainly long tissue, but also bronchial lymph nodes. Most of the TB in elephants is pulmonary. There have been some generalized cases where it's been found in other organs, but certainly the bronchial lymph nodes and the lungs are the main tissues to sample.
From a live elephant.
Oh, from a live elephant.
Yeah.
Oh, okay. Well, other than the trunk wash, it's a respiratory disease. It's going to be in the lungs, so we've got to somehow get a sample from the lungs. There is a bronchoscopic procedure to try to get deeper samples, but that requires anesthesia, and it's not something that would be lightly undertaken. So we need to use respiratory samples that we collect right now by trunk wash. Now I will mention this. We had one case here where the elephant had an unusual skin lesion, and we were able to culture TB from that. And certainly if there would be any oral lesions, anything that looks suspicious, I would culture, but you have to rely on the respiratory samples.
Okay. Any other questions?
Yes, Tsubura Sensei.
Yes.
Can I ask the questions?
Yes, please.
Yeah, thank you, Dr. Mikota, for very great talk about the elephant TB issue. I'm not an elephant or a TB person, but based on your talk, you mentioned about the treatment, and I know it's very, very complicated issue. But I'm wondering that is there any control measures, strategies for prevent the transmission of the elephant TB, like in the captivity environment? And I have another issue concerned the elephant population in the wild. I know you mentioned some cases report in Asia, but I'm wondering, is there any like epidemiology study in the wild for elephant TB? That's my two questions. Thank you.
Okay, the first question was preventing it in captivity transmission. Is that what you asked?
Yes, yes.
Basically, identifying the suspect cases using serology, and then segregating those elephants from other elephants that are serologically non-reactive would be one method to control it. That's probably the best that we can do in captivity. And in terms of the wild, a lot of the work's been done in South Africa. Again, it's M. bovis, so it's a little bit different, although there has been 1 or 2 cases of MTB in African elephants in South Africa as well. And I'm not sure I answered your question. What was your second question?
Yes, is there any epidemiology study in Asian elephant relative to TB, for example, like the mortality rate, the transmission, prevalence, this kind of information?
Well, we've certainly done a lot of work in Nepal, but not exactly from an epidemiological standpoint. It's a little, if we include, and again, in Asia, it's mainly been looking at captive elephants. If we think about looking at the wild in wild populations, it's a little bit more difficult. It's easier to study in Africa because things are more open, whereas we have smaller populations of elephants in Asia. They're in the forest. They're harder to access. I've already presented the captive studies that give us an idea of prevalence among the captive elephants, roughly, somewhere between 20 and 30% of elephants that are in captivity.
Thank you very much.
Okay. Another question is put in the chat box. So any thought on fecal TB testing for elephants? Is that the question?
Fecal testing?
Yeah.
Yeah, that's what, feces are kind of messy. So it can be done, but I think it's a little bit harder to process the samples. And my understanding of TB, humans cough it up and swallow it, and it's excreted in the feces. Elephants don't cough that much, so I think we're less likely to find it in the feces. When we first we're dealing with TB here in the US, our national veterinary diagnostic labs looked at that issue of feces, and they got pretty discouraged with trying to find it that way. So I would think that if you're doing it at all, it's a secondary technique to supplement looking for it in the respiratory secretions.
Okay. I think one comment at the chat box: screening and treating elephant handlers to help prevent spread to elephants.
Yeah, that's really essential, because humans, it's a human form of TB that elephants get. So even though it can go elephant to elephant, once it gets into their populations, we need to screen the humans, and also for their own health as well. So all of the TB guidelines recommend human testing and screening.
Okay. Any other questions?
Thank you for your presenting. As we know, Thailand has many elephant-related tourism activity. I have a question about it. I would like to ask, in your knowledge, do these elephants used for the tourism undergo routine health check, or do they have vaccination schedule every year?
So the tourism elephants in Thailand, I'm sorry, I didn't hear the question.
Hello, can you hear me?
Yeah, I can hear you. Right.
Yes.
You're asking about the tourism elephants.
Yes, I would like to ask, do these tourist elephants, will they have routine health check or vaccination schedule every year?
I think most reputable facilities, yeah, do perform physical exams on elephants and take care of their, any medical issues. As far as screening for TB, I don't really know what goes on in Thailand and whether the tourist facilities are doing that or not.
Okay. So another question is put in the chat box. How often do we need to screen the elephant serologically?
I would think in captive situations, annually would be good, but then if you start getting some reactive elephants, you might want to screen more frequently to see if there's any seroconversions from previously serologically negative elephants, kind of keep a handle on the situation. So annually initially, and then more often as needed.
Okay. Next question. Thank you, Susan, for your great presentation. I'm curious about the breathalyzer. Lastly, you mentioned breathalyzer that detect tuberculosis. So far, this diagnostic tool has only been used in humans. Is there any research with the animal diagnosed with it?
I've been so interested in this for so long. There was actually a company in the UK that I met with we were going to try to adapt their human-developed product to test in elephants, but it just eventually just never happened. There has been some work in cattle. I don't know that there's any licensed diagnostic tests for either cattle and humans, but certainly it's an avenue of investigation that I would love to see someone pursue, because it could be, you know, just so much more economical. You could test them every day. You could test them twice a day with this kind of a technique. So it would be a great research project. And Lasta, I can dig up all the references that I have, and we can do a new lit search. It might be a great PhD project.
Okay. Any other questions? No? But it's almost 1 hour past, so I think we should close the Susan section. Thank you so much, Dr. Susan Mikota.
Okay, well, thank you.
Presentation.
And
Thank
just
you.
a quick hello to Dr. Dekal from Nepal. Nice to see you there.
Thank you, Mikota. I am also glad to see you.
Yeah, nice to see you. Okay, we should close this session. Thank you.
Yes, we can start now.
Okay.